Afternoon, everybody. Hopefully everyone's hearing me okay. My name is Patricia Hart. I'm at Quadram Institute Bioscience, and we have a few presentations from Quadram Institute for you this lunchtime. I'm going to just quickly pop up some housekeeping slides here and share those with you. Okay, so the presentations that we have today, Quadram Institute, for those of you who don't know much about us, has a focus on food, gut health and microbes in the food chain. And we're very interested in keeping people happy and healthy and living longer and healthier as they progress in various different ways. The presentations that we've got today outline how we do that using human studies, how we do that using models of the human gut, and also how we talk about our research and how we use social media in particular to help support that research. And Ruby from our comms team will be talking us through that. Just a couple of quick housekeeping. We are recording this session, and so if you could keep your microphone switched off during the talk, keep your video switched off during the talks, and all the usual caveats about trying to keep phones and so on nice and quiet when we're doing. If you do want to ask a question of any of our speakers, we'll take the questions at the end of each presentation, and we can switch on video and microphone, but if you'd like to type your question into the chat box, we'll keep an eye on that and invite you to speak. So with that then, I will stop sharing my screen and I'll invite my colleague Antonietta Hayhoe to present. She's our Human Studies Coordinator at Quadram Institute, and she's going to tell you a bit about the studies that we're undertaking at Quadram at the moment and how we do that. Thanks, Antonietta.
Thank you, Tricia. Can you just confirm you can see my slide, okay?
I can see your slide, yes, thank you.
That's perfect. So good afternoon, everyone. I'm Antonietta, and I'm the Human Study Lead for the Quadram Institute Bioscience. So first of all, thank you for the invitation to join the seminar today. I will give you a brief overview of the human research that we carry out at QAB. And our research team are working very closely with the local hospital and in particular with colleagues at the clinical research facility that is based at the Quadram and the Norwich Research Park Biorepository team. So at the moment, we have quite a diverse portfolio of projects that they do benefit from a strong relationship with the NIH and other NHS sites. For example, we have the COPS study that is a joint effort from QAB researchers working with the NIH clinician and the team from James Budgett Hospital, recruiting research participants with COVID-19 to assess the prevalence and persistence of the virus in feces. So we have successfully completed recruitment for this study, and my colleague Ruby, in her presentation, she will give you some example of how the social media campaign was quite important for helping the recruitment to the COPS study. At the moment, we are processing and analyzing the samples by using a protocol that was optimized at the start of the pandemic at the Quadram to detect the virus in human feces. In terms of the work that we do with the clinical research facility at QI, they recently have been awarded the NIHR funding. So since October is now the NIHR Norfolk Clinical Research Facility. And so the CRF at the Quadram is one of the three sites. So for us at QAB is really important to work with the CRF. They are it's a really an excellent research environment for food and nutrition studies. We have access to outpatient clinical space and lab space, as well as food preparation areas that are registered with the Food Safety Agency. So, as I mentioned, it's really an amazing environment for carry out nutrition studies. And here I have an example of a nutrition study, the REST study that has been undertaken at the CRF. We actually have delivered all the aspects of this intervention study at the CRF. What was really important from the REST study is that during this project, the team at QAB has validated a remote protocol for monitoring glycemia. So during the first study, the REST, everything was done in-house at the CRF with the research participants coming to the facility and testing the effect of high resistant starch bread on glycememia. Now with this validated protocol, we are undertaking the a remote study, the CLICARB remote, where participants do not have to come to the CRF, but we from the CRF can deliver the study remotely and implementing these remote glucose monitoring protocol. At the moment, we are setting up a new project, so it's called the Harvest Study. The aim is to recruit vegan and vegetarian research participants in a randomized crossover study. We are going to test a new dietary app and an hydroponic unit, so a kitchen garden unit, to alter vitamin B12 and iron status. So we are at the moment getting ready, so we hope that January 2023 we will be ready to have the first participants coming to the CRF and join this interesting project. We already have opened recruitment for the Pearl age. So in this project, we aim to recruit the whole family. So we would like to recruit research participants from at least 3 generations of the same family, with the idea of characterizing the microbiome in the host families. So it's looking at microbes transfers within families. For this project, it is important to work closely with the CRF, but also with the pediatric team at NIH. And of course, we have to really use all our recruitment tools to recruit from across Norfolk, because we would like to recruit at least 60 families across Norfolk. In terms of the work that the human research that we do with the Norwich Research Park Biorepository. Again, we have quite few open studies at the moment. The NRP Biorepository is instrumental for us because we can access fresh and banked human tissue samples, not only from patient, but also from healthy donor. We have the possibility to access samples with the full informed consent from the donor for research. So this is very important. And we have the possibility to design a prospective collection to really help our research. For example, Stefano at the QAB at the moment is leading this project, the PARMIC study, where he can access fecal samples as well as urine and saliva samples from patients with Parkinson's disease. So the idea is to identify Parkinson-specific microbiota, so looking at the microbiome at different stage of the Parkinson's disease. So the PARMIC study is one of many studies at the moment that are funded by the QAB Clinical Seed Corn Fund. So this is really trying, this funding scheme is new, and this is the first studies to be supported by this scheme, and it is really trying to help the collaboration between researchers at QIB and clinician. With the help of the biorepository team, we have established a regular collection pathway of fresh tissue biopsies that are collected when patients are attending a routine appointment at the endoscopy that is based at the Quadram Institute. We have this project at the moment, Mucosodome, that in the last few months has collected more than 300 fresh biopsies using this collection pathway. So the clinician, with the help of the biorepository team, do identify, approach, and consent eligible patient in endoscopy, and fresh material. As soon as the biopsy are collected, they are immediately transferred to our labs at QIB. So as you can imagine, we are really using here high quality samples. And we also have the possibility to access metadata. So there is an extensive metadata coming with the samples. So it's really creating a valuable collection of tissue and data. Via the biorepository, we also access clinical samples, surplus clinical samples. So, of course, these are diagnostic samples that they're primarily used for clinical care, but with the leftover, the remaining materials can actually be used for research under the biorepository ethics. So we have several projects using surplus samples for research, and as an example here, I have a slide describing the work that Nicole is doing at the Quadram, accessing Campylobacter positive fecal samples, so diagnostic samples, and then are shared with our lab to develop a sequencing method that will help characterizing pathogens in fecal specimens. With the biorepository, we also have the possibility to manage the samples and data that we collect as part of our human study using the Achiever Medical, that is a LIMS system. For example, the PEARL and the MOTION studies, they are two longitudinal studies that in the last 4 years have collected a wide variety of biological material from two big cohorts. So we are using Achiever for both studies, and is really important and facilitating the collaboration within QIB, but also externally. And also, this is like the use of this sort of system is gold standards for compliance with the Human Tissue Authority, with the Human Tissue Act. So it's very important for us to have access to these systems. So this was just really an overview of the studies that are open at the moment at QIB. As you can imagine, because we are recruiting research participants mainly across Norfolk at different stage of life, we need to have a diverse strategy for recruitment purposes. And we have a participant database at QIB that does really help recruiting across Norfolk. And as Ruby will go through her presentation, social media do play an important role for boosting awareness of our human research and helping recruitment to these sort of studies. Unfortunately, because of time, I could not give in many details of each project. So if you are interested in looking at the protocols of this project, you can access via clinicaltrial.gov the protocols. All our QIB human studies are registered in this public accessible database, so you will be able to find all the information. But if you also can contact me, and I will put you in contact with the relevant study team. So thank you. As Tricia say, we can take question just after the talk. So I'm happy to take question now or via email offline, whatever is easier for everyone. Thank you, Tricia.
Thank you very much, Antonietta. That was absolutely grand. There aren't any questions in the chat. If anybody would like to raise their hand, I'd be happy to take questions. I'll get things going with, if there are folks listening, Antonietta, who are thinking about setting up a human study as part of their research, but haven't done so in the past or have no experience of it, what would you say the sort of top considerations are in terms of what does a person need to do in setting up a human study?
I think one of the first things to consider is timeline. Most research projects, they would require time in terms of setting up and getting ready in terms of approvals and also in terms of logistics. So what I usually recommend people is really to have a clear understanding of how long it takes the process, because sometimes it's underestimate the time that is required to go through each step. So that is an important starting point. Have a clear idea of your milestone. And of course, I mean, in particular within the Norwich Research Park, every organization will have a support team, a way to just initiate the process.
So get in touch with your local support person then. That's a good top tip. Anne-Marie, you have your hand up.
Yeah, thanks, Trish. Really nice talk, Antonietta. Thank you so much. Just the endoscopy unit is indeed an absolutely fantastic resource, which you highlighted. And there's so many possibilities, I think, for projects that could be initiated with the support of the endoscopy unit. What kind of is the process if one has an idea to kind of, you know, how does one get an idea going if you have one?
So the collection at the via the endoscopy unit is facilitated by the biorepository team, and so is under their ethics as well. So the first things to do, it will be to contact the biorepository to have an idea whether the type of material that is needed for that project is actually collected by, because every study would have different requirements. And in terms of the process, there is what is called the Biorepository Access Committee that every month does meet and looks at new application. So if you have a new idea, you can just complete the application, give all the relevant information, and every month there is a deadline for the committee to accept the application and then review them. So once the application is approved, then the biorepository team will arrange the collection for you. So this is how we are doing at the moment. We got approval for one of the project, as example, the mucosodome, and they are collecting every week. So that is the beauty of the system, because it's a regular collection. So you can plan your experiment and every week access fresh material.
Yeah, brilliant. Thank you very much.
Super. Okay, well, all our speakers are staying with us. So for now, I'll just hand over to Ruby, but if people have questions that pop up as we go along, then we can always come back to the other two speakers at the end. So Ruby, over to you. Thank you very much.
Hello, can you see and hear everything okay?
Perfectly, thank you.
Thanks, Tricia. Yeah, hi everyone, I'm Ruby. I'm from the Quadram Institute Communications team, and I'll be talking about how social media can help research. So why considering using social media for research and the benefits it can bring, some tips, and then also linking back to, as Antonietta mentioned, how we've used social media at the Quadram Institute to help recruit participants for the COP study. So there's several benefits to researchers for using social media. So one of it is it's a great way to get feedback on research. So if you've got a paper coming out or develop a new method, to find out what others think of that and hear their suggestions. It's also a great way to hear about opportunities, whether it's collaborations or learning, things like training, things like that. And also for recruitment, whether that's recruiting roles related to your research or participants, which we'll talk more about in the COPS example. It's also a great way to support your organisation or institute, and also the collective NIHA as well, by helping amplify messages from the different organisations. And so all of these come into that it basically is a great way to broaden the audience of your research and connect with people that you might not meet in person, but meet online. So an important aspect to think about if you're using social media to help your research is who in particular you're looking to engage with. So you may already be aware that there's a variety of social media channels that you can see here, and these have different audiences. So it's important to think about who you're looking to engage with and which might be the most appropriate channel. So Twitter, as you may be familiar with, is kind of probably the most academic of the channels, so where most academic discussions go on. But it's also used a lot by policymakers, so MPs and people like that. So if that's the kind of audience you're looking to engage with, that's a good channel to consider. And also just to mention that journalists as well are often quite active on Twitter and might put out, for example, when they're looking for comments or expertise on a particular topic. Facebook tends to be kind of more for local things going on, because people tend to be connected more with their friends and family. So that could be a good way, perhaps if there's events going on, like public engagement events, to connect with people in that way. LinkedIn is kind of more industry focused and jobs and careers. So if in your research you're looking to connect with perhaps new industry partners or things like that, that can be a good place to do that. Instagram and TikTok are kind of more visual based and tend to have younger audiences. So for example, perhaps if your research is more visual based, like microscopy, Instagram can be a good place to consider engaging with people. And TikTok has the youngest audience of all of them. So perhaps if you're thinking to inspire the younger generation, that might be a place to consider. So you might be thinking, what do I have to share about my research online? So as mentioned before, papers and things like that is a great way to share online. Also, yeah, research, preprints is a great way to share online, so you can get feedback on your research before it's published. Also, public engagement. So it's a great way if you're attending public engagement events to let people know that you'll be there in advance, but also to perhaps continue discussions after events. So perhaps if you meet someone at a public engagement event and have a dialogue with them and they have a follow-up question, and then you can perhaps continue that dialogue afterwards. And also, if you're attending conferences or presenting posters, if you share that online, it could be a way to broaden your audience for people that haven't attended that particular conference. So you can see here an example of one of our researchers, Emma, from Quadram, sharing work from the language group, their work, and so helping to broaden the reach of their research. Also, as mentioned before, it's a great way to support all the organisations, as well as NIHA collectively, so by engaging with posts and sharing them, and also tagging as well, if the institutes, if you're talking about their work. So some practical tips if you're considering using social media for your research. So you might already be aware of these too, but images are a great way to engage people and grab people's attention online. So you can see here that there's evidence suggesting that when you do use images in posts, that it engages lots of people. Also, just to mention that alt text is a great thing to include with images. So you can see here on the right an example of that. So that's basically a description of what's in the image, and that's particularly good for accessibility. So if people are using screen readers, if they have visual impairments, it helps make what's in the image accessible. And also if people live in areas with maybe weaker internet, so the images don't necessarily load, but you can see what the image, you can hear or see, read what the image shows. And also just to mention that obviously it's good to get people's consent if they are featuring images. So just verbally to check with them. And also, as sort of mentioned, tagging is a great way to help broaden the reach of your online presence. So this lets people know if you're mentioning them, and they can encourage them to share more widely too. An important aspect, I think, to consider if you're thinking about using social media for research is frequency. So if you're looking to share a particular piece of news or research, but not looking to engage kind of with social media more regularly, it might be more appropriate to contact your organization's communications team for them to share via their channels, rather than setting up an account just for perhaps sharing one particular thing, and then it might become out of date. Hashtags are also a great way to contribute to wider discussions that are going online. So you can see here again in this example, sort of contributing what's going on to a wider discussion. These tend to be things like awareness days or awareness months, but other things too. So it can help make your post more discoverable to a wider audience. And also just to mention, it's good to be aware of each organisation's social media policy, which are usually available on things like intranets and things like that. So moving on to the example which Antonietta has kind of mentioned about the COP study. So this is an example where, as an institute, we use social media to help support the research by recruiting participants. So the aim of the campaign was to recruit 100 eligible study participants who are over 18 within 40 miles of Norwich, and had had COVID in the last two weeks. So here you can see what the campaign looked like. So using things like images to engage people. And also to mention the social media was, as well as organic posts, which is where you just post directly on social media platforms. There was also a paid campaign, so where you pay for the post to be promoted, so they appear on people's social media feeds. And what's quite helpful about that as well is that you can target campaigns, so to a particular geography, so in this case within 40 miles of Norwich. So here you can see the results of the campaign, which proved successful for helping to recruit participants. So in the top right, you can see just over 30,000 people saw the posts across social media, and from that, people engaged with it and then went to the web page. So on the left, you can see how many people viewed the web page, and also on the graph here, kind of when there'd been those paid campaigns that I mentioned, how it led to an increase in people looking at the web page. And then from that, people filling in the form to show that they're interested in taking part, and proved successful as it met the target of 100 people being recruited to the study. And also you can see below how from Twitter that there was a wide sort of distribution across Norfolk of people that saw the post online, and also across ages. So it proved a really useful tool in helping to recruit people to the study, and along with complementing other ways as well of recruitment that Antonietta mentioned. So for example, as well, you can see here, so the EDP featured the call for participants, and that also helped contribute to, but social media proved a valuable tool. And also just to mention as well that all the campaign materials kind of reviewed ethically internally and in NHS. So if you are thinking about recruiting participants, social media can be a great way to support that recruitment. So thank you very much for listening. You can find here some more links to information about how using social media can help research. And yeah, if you have any questions, let me know. Thanks very much.
Thank you very much, Ruby. That's great. And well done for surviving the Zoom bomb. So does anybody have any questions? There's nothing in the chat box at the moment. I have a quick question. Well, I think it could be quick. It probably, there's a lot more to it, but you sort of mentioned the sort of top tips for social media, but what are the sort of main pitfalls that people might want to consider if they're using it in their professional capacity?
I think one of the things, images, where I kind of mentioned resolution, that can be a thing you definitely see. It's good to make sure that they're highest quality as possible, and yeah, it's great to tag people, I think, as well. It's a great way to kind of help broaden the reach of your audience, and also tagging organisations and institutes to help encourage them to share your research that you're sharing online.
And most organisations, I mean, most of the organisations that are part of NIHA, they would have social media officers like yourself, or that people can go and talk to, or not necessarily? Yeah, I think most of them do, don't they? No, comms teams at least, right? Excellent. Okay, well, again, Ruby will be around for a little bit longer, and I'm sure we'd be happy to talk to you. Yes.
Question from David there. He's got his hand up.
Oh, yeah. Sorry, that hasn't appeared. Right. Okay. Yes. Go ahead, David.
Hi there. Thank you so much, and thank you for a great talk, Ruby. It was really interesting to hear the sort of methods that you're using for recruiting and publicising your research. I was just wondering, because I'm using some social media for my PhD research, but ethically I'm unable to recruit myself. I need to go via sort of a charitable organisation. Have you got any considerations around the ethics of online recruitment, and how sort of an individual such as a PhD student might be able to influence an organisation to employ some of the methods that you've said there around sort of promoted tweets and things?
So with the ethics, I'm not sure if Antonietta might be able to talk perhaps more about that.
I think what perhaps practically what we always do when we have a new study and we wanted to use social media, and as you say, sometimes is also in our case, we are not directly initiating that recruitment. We have to rely on someone else. What we found very useful to make sure that ethically everything is covered is actually to submit to the ethics committee the full recruitment strategy with actually all the text that is going to be used, where, by who, say everything that Ruby has explained actually been part of a document that is the social media campaign material, by saying also the frequency, the people that are going to be responsible for the different channels. So in that way, we are sure that an ethics committee has looked at it, has considered if there is any ethical issue with that, and so we feel confident that everything is done okay. And the other things to consider is, before actually going to the step of contacting the ethics committee for review, it's actually good to talk with the organization that you're working with. They will have some people that are responsible as well for what they can and they cannot advertise. So I think in the engagement at the start is a good idea, and then put everything in black and white for an ethics committee to really have a full understanding of what you're going to do. Does that answer your question?
Yes, thank you. That's really helpful. Thanks.
Brilliant. Thank you very much, David, for that question. Fab. Okay, I don't see any other hands, so hopefully that was the only one I missed, so apologies, David. Thank you very much again, Ruby, and thank you again, Antonietta, for that input as well. I'm now going to hand over to Lee Kellingray, who is one of our research scientists at Quadram and has spent recent years building up what is a really interesting and very fascinating model colon centre, because not always are bodies available for us, and it's always good to have a look in vitro first. So I'm going to hand over to Lee to tell us a little bit about what he's been up to. Thank you, Lee.
Okay, so it's disappeared. Oh, there we go. Disappeared. Also, sorry, I'm in the dark. I was completely dazzled by sunlight, so I don't normally dwell in the dark quite as much as it seems. Is that the correct screen?
We've got your your presenter view at the moment.
No,
that's better.
Okay, so I'm going to talk to you today about the QIB colon model facilities, where we have 3 models which we use to simulate the human gut, and these are particularly useful for testing dietary compounds, pharmaceuticals, prebiotics and probiotics, but they can be used for a variety of uses. The model which we've used for the longest, called the Gibson models, this is due to them being designed by Glenn Gibson at the University of Reading. And these were designed in the 1980s, but still work perfectly well, as my boss tells me, and he's in his 60s, just as old, it doesn't mean it's useless. The setup we have here is what we call a batch model. This is a self-contained model where we don't add fresh media during the experiment. And due to that, we tend to run them for between 24 and 48 hours. Otherwise, you get a buildup of bacterial waste products and depletion of nutrients, which therefore means it no longer reflects what you'd expect in the human testing. 2 different sized vessels as well, with working volumes of 150 mils and 50 mils. I'll just zoom in and sort of explain some of the components of it to make it a bit more understandable. Towards the top there, we have a line to nitrogen cylinder, and this passes through a filter and is constantly bubbled in at a slow rate to create an anaerobic air environment. Towards the back, which is quite difficult to see, but you might see a filter there. We have a small bit of tubing with a filter which is inverted. This allows the release of gas and the release of pressure, but we add the filter so we're not releasing bacteria into the working air environment. The glass models have a water jacket, and these are connected to a water bath to maintain a temperature of 37 degrees. The media that we use was specifically designed to simulate the intestinal milieu. And in fact, it was designed by taking samples from people who had suddenly died. It was a collaboration with a morgue. So they really went to some effort, and it was a little bit, oh, actually, I shouldn't be in the dark when saying that, it makes it spookier. But so that's been a validated media. The vessels are on a stirrer to mimic peristaltic movement. And we have a sampling port because it's important to be able to take samples quickly so as not to disturb the experimental air environment. We also have a pH probe which constantly takes measurements of the pH whilst the experiment continues. And this is connected to a pH unit where you can set the pH where we have it set between 6.6 and 7 to mimic the human large intestine. And when the pH falls out of this range, the appropriate pump for acid or alkali will kick in. And the tubing goes from a weak acid and alkali through the pump, and it attaches to the pH feeder just up the top that looks a little bit like a glass cactus, and that gently drops the acid or alkali in until it brings it to the pH range that we are looking for. As I say, that's the batch model. It is possible to set them up in a slightly different way, but with all the same components, to form what we call a continuous model, where each of the 3 vessels mimics the ascending, transverse, and descending colon, and the pHs are set to different pHs corresponding. The main difference is that whereas the batch model is a closed air environment, with the continuous model, you have a large volume of sterilized media, which comes out through the tube and through the pump, which the pump is set to a particular flow rate with a calculation, which means you can, in essence, model normal bowel constipation or diarrhea. And this flows through the vessels and then out into a waste pod. Normally, this would be run for approximately 2 weeks to allow the microbiota to stabilize once it's been seeded from a human fecal sample, which we obtained from an ethically approved study called QID colon model study. There are a list of participants who are willing to donate samples on occasion when asked. Due to this being a continuous model, we don't have to build up with waste products or the depletion of nutrients, so these can generally be run for up to a month and a half. The Gibson models were created in the 1980s, and a few years ago, just before the pandemic, we got a BBSI SRC grant to buy a compact high throughput colon model system called the Micro Matrix. It has a smaller footprint than 3 of the classic batch models, yet it works via a 24 well cassette. So in essence, for the space of 3 Gibson models, you can have 24 small independent bioreactors. And each of the wells, which have a working volume of 5 mils, at the bottom of the wells, they have a sensor to take pH readings, one for temperature and another for dissolved oxygen. And when you set up the software, you indicate what range you would like for dissolved oxygen, pH, and temperature. And the pH is altered not by liquids in this case, but by the addition of carbon dioxide or ammonia gas, which once again is filtered, as is the release of gas. This means the volume doesn't change, which is very important if you're working with small volumes of 5 mils. And one of the main advantages of this particular piece of equipment is it's incredibly quick and easy to set up compared to the previous models. But also, due to having 24 wells, in essence, with one fecal sample, you can run an entire experiment with enough replicates for publication, and therefore you aren't restricted by the variability that comes from different fecal samples from the same people, even a day later. And this has been used well so far. Our most recent model, considering we've gone from small and compact, is a bit of a beast, but it is at the moment possibly the most well-known model for validated human intestinal models. And it's called the SHINE model, a simulated human intestinal microbial ecosystem. And although in this picture we're showing 5 vessels, it can run up to 10 vessels at a time. And once again, you have acid and base, you have a water bath and pH probes, and these are all regulated by an inbuilt computer. And due to its modular nature, I mean, all of these little outcroppings you can see are pumps. And due to the modular nature, it's very flexible, which is a great asset considering how complex the human gut microbiota is. This model is also not just restricted to the colon. it's possible to set it up in such a way to focus on the upper gastrointestinal tract, and it's also possible to use dialysis within the experiment as well, which gives it greater specificity to the research question. Alongside that, it can also perform short batch models or long-term continuous experiments based on the colon. And it can also be used to simulate the small intestine, which is a great asset to this machine, because not a lot of the colon model systems that are out there are able to do this reproducibly. I'm going to run through a few different setups. So, as I say, it's modular, so you can you can set the experiment up to really focus on the main point of your question. The twin shine, in essence, twin just means that it models a simulation of two individuals. So, for instance, for individual 1, we would have a stomach vessel and a small intestinal vessel, same for individual 2, and then you'd have the ascending colon, transverse and descending colon. And once again, this is a continuous model, so that you then have a waste pot. If you're not so interested in what happens in the particular sections of the colon, it's possible to run the triple shine model, which simulates 3 individuals. And the only differences there are it's run in such a way that the stomach and small intestinal vessels are a single vessel. And the colon, instead of being split into 3, is split into proximal and distal regions. This allows you to do more replicates again with a single experiment. Regards to the upper gastrointestinal tract, although it's showing 3 here, you can run 5 of these sets at a time. And the idea is that once you've performed your experiment, which you can simulate fasted or fed, which is useful for glycemic response, for instance. And the material that you get at the end of this experiment, you then have the option to use that material for a colon model experiment to look at the entire gastrointestinal tract. But we're more focused on the upper GI. This is a bit wordy, and I wasn't entirely sure of keeping it in, but I just want to give you a general idea of what happens when the experiment is actually running. So this is all automated, so gone are the days of turning up at 2 a.m. And from the first day to the final day, it simulates 3 feeding events to simulate breakfast, lunch, and dinner. Dinner and tea, depending on what region of the UK you're from. In essence, it starts off by adding the nutritional medium and pepsin to the stomach after a certain amount of time, which is purely programmed in, depending on your research question. This is then transferred to the small intestine, where pancreatic juice is added. And then following that, it's a series of transfers from small intestine to ascending colon, transverse, descending, and then to the waste pot. As I say, this is all automated, which is not something that you get with the Griffin models. An example of a typical experiment, and in this case, focused on a prebiotic, is that you would have a stabilization period. And this is where at week 0, you would obtain a fecal sample, in our case from our QIB human study of colon model participants. And then you would allow that up to 2 weeks for the microbial community to stabilize. And this is tested by intersecting the material that comes from the descending colon to the waste pod and testing that for short chain fatty acids, as that gives a very good idea of metabolism and bacterial activity. So once the short chain fatty acid levels have become stable, you can infer that your microbial community is stable. Once it reaches a stable point, you then could run it for another couple of weeks as your control. So this is before you've added your substrate of interest, in this case fiber. to know what the bacteria function and composition consists of when it's not changed. And following that, you would have your treatment period, and then obviously from the samples that you take, you can compare your treatment period, your control period, to get an idea of what we would expect would happen in the real world. After the treatment period, you could have a washout period to then to confirm that the microbial community reverts back to the control. So from this experiment, which was performed by ProDigest, who are the makers of Shine, they focused on short-chain fatty acids, and as you can tell, the control period is largely stable. Once the treatment was added, we see an increase in all three of the major short-chain fatty acids of acetic acid, which had the almost immediate response. Butyric acid, which the response was a bit more delayed, but was still quite a large response, particularly compared to propionic acid, which is a more of a measured response, but still an increase. And then following the end of the treatment period, we can see the short-chain fatty acid levels came down again and then remained relatively stable across the 2 weeks. One of the unique selling points of the Shine model is that it's possible to add mucin agar-coated microplasms, which are put in a small netted bag. You have 4 of those netted bags within a larger netted bag, and this allows for to see the effects of your compound of interest on not only the luminal bacteria and microbes, but also those which are more evolutionarily adapted to populate the mucin, which is a very important area which has lacked in some of the more older models, considering how close it is to the intestinal cells and the links that it has, in particular with IBD. Just finally, ProDigest did run a validation experiment of their model, where they took the same human fecal microbiota obtained from the fecal sample and processed, and they added it to the Shine, as well as germ-free rat model. And they did a 3-week treatment with the Ravenose, Xylan, and Inulin to look at whether the effects seen are similar between the in vitro model and the in vivo model. Due to the different volumes of the Shine vessels compared to the rat secum, we first calculated proportional concentration as a percent of the 3 main short-chain fatty acids. And I found fairly remarkably similar profile by the end of the experiment for acetate, propionate, and butyrate between the Shine model and the rat secum, which not only gives confidence that the model that you're using can be trusted, but also it gives a reason why we can reduce the amount of animal work that we do, particularly prior to human studies, which is obviously something that we are all trying to move towards. So just to finish off, the Shine model came from a BBSRC grant last year, and it was led by Natalie Jude alongside myself, Arjun, Mike, and Reg. Arjun was responsible for obtaining the Gibson models a couple of decades ago. People in the central column are from the business team, contracts and finance, and they were particularly helpful in this post-Brexit world, trying to get something from Belgium to England and obtain a contract which was suitable for all parties. Tim and Chloe were my point of contact with ProDigest, and BBSRC obtained a grant, and study donors are the most important, as without them we cannot run colon models So we absolutely rely on them to help us with our research. Thank you for yourselves for listening.
Thank you very much indeed, Lee. We have immediately a question from Julia, who has asked, why is 5% cecal inoculum used in the SHINE model, and is the inoculum added to each of the vessels?
So 5% is largely what ProDigest uses. We generally find 1% is a good proportion for communities which are large and complex, and 5% we generally use for pure cultures. Sorry, what was the second part?
The second part was, is the inoculum added to each of the vessels?
Yes, it is, yes. And then the particular conditions, largely the pH, will be what selects for the community. So you will get a slightly different community in each of the vessels, depending on what settings you've programmed into the machine. But the starting material is the same for each vessel. Thank
you very much. Just so everyone's aware, we're not being Zoom bombed. Lee unfortunately sits next to the kitchenette and it's lunchtime, so Anne-Marie.
And someone's cooking something very nice in the
microwave. Anne-Marie, your hand's up.
Yeah, thanks a lot, Lee. It brings me back to my PhD days when I used to use dialysis tubing and looking at iron absorption. Yeah, the models are really sophisticated these days, and thank you for going through the various options with us. I guess the downside is it kind of always lacks the cellular material of the intestine. So you never have the influence of the gut endothelial cells and the impact that has on microbial digestion and the digestion of the various food components. Is there any option in any of your models to add cellular material to kind of increase its physiological relevance?
So we did look into whether we could convert the Gibson model into more of a transwell model, where you would have a cellular component separated but still permeable to small molecules. And it became quite difficult, but it's certainly going to be the next step. I mean, the group, Natalie Judra's group has gut on the chip, and they also use transwell models. And it really is the next step. We'll really be taking that, infusing it into a particular one. I mean, I'm just waiting for someone to create a robot that has innards that would be similar enough that we could just have one in the lab, but I fear I may not be still working at that point. It's a difficult one, and in particular, even with the transwell, you have the difficulty that, as far as I'm aware, there's not standardized media, which the human cells are happy in and the bacterial cells are happy in. So some work would also need to be done on researching that.
Yeah, thanks a lot.
I love the idea of a robot wandering the corridors, quietly digesting food for us to go back to and sample. That's wonderful. Any other questions for Lee? I'm going to do a shameless plug for Quadram. We do have this series of integrated models of the human gut now, as we call them. So it's all the work that Lee's been talking about, the various different systems. It's the gut on chip model that he just mentioned that Natalie Judra's group is running. They're also expanding that out now to other organs on chips. We've got quite a close collaboration with the company that creates those. And then of course we've got the human studies and the germ-free mouse colony. So I think for anyone who has any ideas across NIHA to do any work that is studying the gut or studying how food is digested, please do come and have a chat to us, because we'd love to talk you through some of the mechanisms that we could provide to help you with that research and to collaborate or to provide a service to you. So are there any other final questions before we wrap up for the afternoon? If not, I'd like to thank Lee, Antonietta and Ruby very much for taking the time to talk to us today. It's been a really nice way of illustrating how there are various multiple components that contribute to a single piece of research. And I'm sure they'd be very happy to talk to you afterwards if there's anything you want to follow up on. I'm just going to quickly run through another couple of items that Anne-Marie and the team would like to share with you. So hopefully these slides are going to be popping up. So the next NIHA seminar will be Plant Foods and Health. That's on the 2nd of December and will be again at 12 noon via Zoom.